Hello, everyone, and welcome to today's keynote webinar titled Exploring the Potential of 3D Organoids Through Live Cell Analysis. This webinar is a part of the Future of Biomedicine, Reproducible Advanced Cell Systems and Translational Insights. I am Kaylee Bach of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by Sartorius. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. And with that, I would like to now welcome today's speaker, Kalpana Barnes, Manager, Cell Imaging Applications at Sartorius. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Kalpana, you may now begin your presentation.
Thank you for joining this webinar on the study of 3D organoids using the IncuCyte live cell analysis system. I'll start with a brief introduction to organoids and their potential applications, describe some existing challenges with their use, and how live cell analysis can help address some of these. Describe IncuCyte validated protocols with a guided setup for organoid culture and assay, and share some data exemplifying how the integration of the IncuCyte system into organoid workflows can facilitate in vitro research with these complex cell models. Organoids are miniature 3D in vitro models that exhibit similar functionality and architecture to their tissue of origin. Established from pluripotent stem cells, which include embryonic or induced, or tissue-resident adult stem cells. They are grown within a three-dimensional extracellular matrix, often matrigel, and defined culture media. And together these support organoid formation and maturation. Capable of self-renewal and organization, they are amenable to long-term culture while remaining genetically stable. Cultures do, however, lack specialized cell types such as stromal and immune cells, and lack vasculature. Despite some of these biological limitations, organoid technology offers great potential to accurately model organ development and human disease, and provide platforms for drug development, personalized and regenerative medicine across a range of therapeutic areas. There are some practical and analytical challenges with organoid cultures which still limit their potential and accessibility to researchers. The lack of standardized and validated protocols, poor reproducibility and poor compatibility with automation, high costs and low throughput are some factors which can hinder their adoption, especially within a drug discovery setting. Data capture and analysis from these complex heterogeneous cultures and interpretation of multiparametric data can be daunting. Data acquisition and analysis approaches can often be bottlenecks to extracting meaningful data from organoid cultures. The ability to make objective, unbiased decisions about data will further support the validity of organoid applications. Live cell analysis, defined simply as continuous cellular measurements, can address some of these challenges. Systems such as the IncuCyte, which sits within a cell culture incubator, are ideally suited for this. Using time-lapse imaging that spans hours to weeks, real-time continuous monitoring and quantification of dynamic cellular behavior from living cells is achieved. The concept is applicable to the whole cellular workflow, from checking cells during the culture phase through to evaluating their manipulation prior to functional assessment throughout the whole time course of an assay. The system can be used to study and address biological questions related to cell proliferation, cell function, movement, and cell morphology. This schematic describes at a high level key phases of the organoid workflow. The IncuCyte system is ideally suited to support both culture and assay phases. Once stem cells are isolated and transferred into matrigel, cultures can be automatically imaged and analysed during routine culture in 24 or 48 well plates. For assay in 96 well format, any modifications made to organoids, for example genetic or treatment perturbations, can also be tracked and quantified using the IncuSite Organoid software module. This automated approach provides a standardized and integrated workflow from organoid generation and culture maintenance to final assay with image-based, label-free measurements, all within the relevant environment of your cell culture incubator. Here we describe the IncuSite validated protocol and guided setup for culturing organoids embedded in Matrigel. Once harvested, organoids are resuspended in Matrigel and the suspension added as droplets to either 24 or 48 well microtiter plates. After incubation at 37 degrees to polymerize the Matrigel, droplets appear as domes and can be overlaid with culture media. Images can be acquired within the IncuSite system to automatically visualize and quantify phenotypic parameters from all organoids within the dome. Here are some IncuSite videos capturing in real time the formation and growth of mouse intestinal and pancreatic organoids in 24 well plates. A novel bright field image capture approach returns in focus 2D images of all individual organoids distributed and embedded throughout the whole Matrigel dome. This first visualization is of an uncorrected IncuSite image where image illumination and stitching artifacts are visible. Note the dark well edges and grids in the videos. Upon processing, these artifacts are cleaned up and resolved, as seen with the corrected images. Note the robust and accurate segmentation of both organoid cell types, which exhibit very different morphologies. The time courses of bright field area reveal cell type specific growth profiles. The IncuSite automated image acquisition and analysis software can be used to remotely monitor the development of your cultures and experiment. Using the microplate vessel view, you can rapidly and simultaneously visualize images from all wells, and the zoom-in function allows you to interrogate images more closely. Here, the microplate vessel view and time course plots show organoid growth rate and size is proportional to cell number. This automated approach lends itself to a range of alternative matrix experiments, such as defining culture media components and growth factors for optimal organoid formation, growth and maturation, all based on temporal phenotypic measurements of organoid size. Here are some example images of a range of mouse organoids and associated quantification. On the top panel are the automatically acquired bright field images of all organoids within the Matrigel dome. In the middle, the outline segmentation masks, which accurately segment organoids of different morphologies, and on the bottom, the real-time analysis of organoid area. The combined images and time courses reveal cell type-specific morphological features and growth kinetics, with pancreatic and hepatic organoids exhibiting similar morphologies, rapid growth rates and size. In contrast, the intestinal organoids appear smaller, show a distinctive budding morphology as they mature, and have slower growth kinetics. IncuSite metrics can be used to establish optimal organoid culture conditions. Defining optimal culture conditions and regimes is not only key for maintaining, expanding, and cryopreserving organoids, but also for ensuring that cultures are consistently prepared for downstream assays. Integrated quantitative organoid size and count metrics enable you to objectively define such conditions. Here, bright field images and time courses illustrate seeding density-dependent growth. Cultures seeded at too high a density result in overcrowding, and you can see beyond 144 hours the average size of organoids declines. Organoid passage in frequency is driven by indicators of maturation such as size, morphology, and accumulation of debris within a lumen. Hepatic organoids are often described as not being ready for passage when they exhibit clear lumens and are approximately less than 100 microns in diameter. Here, representative bright field images shown 2 days post-seeding indicate that cultures are not ready for passaging, since organoids are still small and exhibit clear lumens. A decline in the eccentricity metric, which describes how round the objects are, is observed within 48 hours as organoids form and become rounder. Over time, as the organoids mature and increase in size, a few in the culture turn black. These cultures are described as ready for passage. Here, the optimal period for passaging occurred between days 4 and 5, when organoids within the dome had reached maximal size, have a rounded morphology, and have not collapsed. Organoids have exceeded their passage window once lumens of most have darkened and many have collapsed. Images and time courses beyond day 6 show a marked increase in the darkness and eccentricity metrics as organoids darken, collapse, and lose their round shape. Intestinal organoids exhibit a more irregular morphology and typically accumulate debris within their lumen. The extent of budding and debris accumulation is used to determine passaging period. The eccentricity metric can be used to track the degree of budding as maturing organoids transition from round to an irregular shape, and the darkness metric to track lumen debris accumulation, with the optimal period occurring at day 7, as the eccentricity metric begins to plateau and most of the organoids have budded and have darkened lumens. The IncuSight live cell imaging and analysis approach supports robust assessment of long-term organoid expansion. The bright field images shown here demonstrate maintenance of the distinct budding morphology of intestinal organoids across multiple passages. The metrics show when maintained at a consistent density, intestinal organoids exhibit comparable count, area, eccentricity, and darkness measurements across passages. Hopefully, having demonstrated how live cell analysis can facilitate the kinetic assessment and quantification of key parameters associated with routine culture of organoids, the remainder of the webinar will focus on how live cell analysis can also support and be applied to accelerate and support organoid research and discovery. Here we show the validated organoid IncuSight assay protocol, still using organoids embedded in matrigel. However, now the organoid matrigel suspension is seeded into 96-well plates using one of 3 validated formats, either as a 10 microliter droplet as described in the culturing setting, or directly to the assay plate, or on top of a layer of polymerized matrigel. Choice of format is dictated by the health of the organoids and organoid morphology as visualized in the culturing phase of the organoid workflow. In this example, we can see that for intestinal organoids, only when seeded as a dome in 96-well plates, organoid size, morphology, and health of cultures at day 7 align with data from the culture setting. However, for hepatic organoids, all formats are conducive for assay, with cost or ease of preparation defining choice of format. All suspensions here contain 50% matrigel. Here we show some example IncuSight videos of organoids in 2 different 96-well formats, either as a dome or layered cultures. Note the consistent capture of in-focus 2D images of different morphologies and textures. To further demonstrate the software's ability to distinguish organoid morphological differences, human brain organoids derived from healthy or epilepsy iPSCs were embedded in 50% matrigel and imaged over 8 days. The images and quantification show that both display comparable growth with a similar average area, but show distinctive morphologies, with healthy organoids being darker and more rounded with a lower eccentricity value, and patient organoids being looser and more disorganized with a higher eccentricity value. Together, the images and associated metrics can be used to study disease and support drug development. Quantification of organoid growth and death can be achieved in 96-well plates. The videos and time courses show the different temporal profiles of both intestinal and hepatic organoids in 2 assay formats, tracking not only their growth but lack of growth, here induced by the treatment of a cytotoxic compound, staurosporine. Growth kinetics and morphology metrics can be combined to quantify responses to compound treatment. In this example, hepatic organoids treated with staurosporine continue to grow and increase in size and number over time, while those treated with staurosporine show a marked reduction in both of these metrics. The zoomed-in images and time courses also show a simultaneous increase in organoid eccentricity as they lose their distinctive round morphology, and an increase in darkness as the organoids collapse over time. Since patient-derived organoids preserve the morphological and molecular characteristics of the tissue or tumour of origin, they are increasingly being used as in vitro drug development models. The ability to distinguish between and quantify cytotoxic and cytostatic cellular responses is important to establishing effective anti-cancer therapies. To exemplify this drug-specific effect, hepatic organoids were treated with chemotherapeutic agents of different mechanisms of action. The representative bright field images show compound-specific effects on organoid size and morphology, and the concentration response curves constructed from the area under curve analysis of organoid area, eccentricity, and darkness time courses demonstrate the difference in profiles between cytotoxic and cytostatic compounds. The known cytotoxic agents, staurosporine, shown in pink, and cisplatin in teal, attenuate organoid growth, compromise and break down the uniformly rounded morphology of hepatic organoids, reported as a concentration-dependent increase in the eccentricity metric, and cause a concentration-dependent increase in the darkness metric as the organoids die. 5-FU in black, while a potent inhibitor of organoid growth, can be described as cytostatic. The compound has little or no effect on organoid morphology or inducing organoid death, as reported by the eccentricity and darkness metrics respectively. In combination, these three label-free metrics can be effectively used to probe and classify compound mechanism of action. CFTR patient-derived organoids are being used to aid functional studies, drug development, and personalized medicine strategies in cystic fibrosis patients. Reports show increases in cyclic AMP levels cause rapid swelling of intestinal organoids by opening the CFTR channel, and that this response is lost in organoids derived from cystic fibrosis patients. Here we demonstrate a label-free, non-fluorescence-based approach to kinetically visualize and quantify CFTR function. Mouse intestinal organoids formed for 3 days were treated with increasing concentrations of forskolin and imaged every 15 to 20 minutes for up to 7 hours. Quantification of the bright field images show that following forskolin treatment, organoids increase in size, become more rounded, shown as a decrease in the eccentricity metric, and display clear lumens, shown as a decrease in the darkness metric. To demonstrate that forskolin-induced swelling is CFTR dependent, organoids were pre-incubated with the CFTR inhibitor 172 for 2 hours. Concentration response curves show that forskolin-induced increases in organoid size were inhibited by this agent by approximately 60%. In summary, the IncuCyte live cell analysis system provides purpose-built solutions for 3D organoid workflows from culture to assay. An easy-to-use guided software interface can be accessed remotely, providing a setup and walk-away experience for users. Established protocols and integrated automated software enable kinetic assessment and analysis of morphological features for organoid cultures without the use of fluorescence. And within an assay paradigm, label-free, multi-parametric data can be used to drive objective decision-making and ultimately better predict therapeutic outcomes. Additional information and what has been described, including more about the IncuCyte system, organoid-specific software and applications, can be found on our website. You'll also find articles and additional webinars from others using IncuCyte live cell analysis solutions for their organoid research.
All right. Well, thank you, Kalpana, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Submit your questions in the Ask a Question box located on the bottom of your screen. All right, so let's get started. We've already got some great questions coming in here. Kalpana, this first question asks, can I image 6-well or 12-well plates using this module?
Okay, great question. Currently, we only support both 48-well and 24-well microtiter plates for the organoid QC approach that I described. So that's for culturing and maintaining your organoids. And we support the 96-well plate for organoids that you take forward into an assay setting. So not right now for 6-well or 12-well plates.
Great, thank you. Our next question here asks, can I spot multiple domes in a single well?
I would recommend that you don't spot multiple domes in a well and just stick to a single dome within a well. That way, your plate is set up for optimal imaging from the IncuSite, which will capture all of the organoids that are present within that single dome.
Great, thank you. All right. Next question here. Can I image fluorescently labeling organoids?
Great question, and a big ask from a lot of listeners. So right now we don't support the ability to capture any fluorescence from organoids, whether in the culture phase of the workflow or the assay phase of the workflow. We support the label-free measurements as I described in the webinar.
Great, thank you. All right. Now this next question asks, can I perform a 96W embedded assay using the Organoid QC module?
Okay, so I think what the user's trying to do there is use the 96-well plate using the Organoid QC software module. So no, the QC module is specifically set up for 24 and 48 well plates, and the assay module is designed specifically for 96 well plates.
Great, thank you. Another question here. Can I harvest the organoids if I want to cryopreserve them?
Most definitely from the culture phase of the workflow. So there, where you are growing them, maturing them, and expanding them, you can most certainly harvest them and cryopreserve them, as described with a lot of literature references of how to do that. So yeah, most definitely.
Great. Now, how do I know which organoid assay format option to use?
Yeah, so you saw that we support 3 validated formats, and what I would recommend is that you select the one in which the growth of your organoids and their morphology aligns to what you see in the culture phase of the workflow. And I think the example I described is for the intestinal organoids, we had to set that up as a droplet or a dome setup. But for the hepatic and indeed pancreatic organoids, which we've tested, you could use all of the formats described. And I think the one you would choose in that case would be one that's easy to set up. So just on a layer would be the most straightforward. And also it be, if you were choosing the dome setup, that would be the most cost effective, requiring very little matrix gel in your experiment. So hopefully that answers the question.
Great, thank you. All right, another question we received here. Can organoids grow from one type of cells in the matrigel, such as one cancer cell line? And how can you isolate cell from gel for downstream analysis?
Okay, so yes, is the answer to that question. We have experience and have set up cultures using fragments, so a fragment digestion of your organoid culture, and then seeded that into matrix gel, for example, in 96 well plates for downstream assays. Or indeed, we've performed digestion of the organoid prep down to single cells and then shown that they grow from a single cell, whether that's in the culture setting or in the assay setting. So in terms of harvesting the cells, there are agents that are available to do that from 96 well plates. And we've indeed harvested them and looked at marker expression, for example, using flow cytometry. So looking at differentiation markers of organoids, as I said, using flow cytometry as a tool. So yes, indeed, that is possible to do that.
Great, thank you. We're getting some great questions in from our audience still, so we'll continue with a few more. This next question asks, what if I want to fluorescently label the organoids for markers? Is that feasible?
So yes, I would say that is feasible. And again, we have done that, as I said, in the flow cytometry setting, but we have harvested and extracted those organoids from the matrix gel itself. And there are lots of agents that are available from a whole host of vendors to allow you to do that. So yeah, that should be possible. And just to reiterate, we won't be able to look at any fluorescent metrics using the IncuCyte Organoid QC or IncuCyte Organoid assay software that was described.
Great, thank you. All right, next question here. How do I minimize matrigel droplets or domes spreading out across the well?
Okay, so that's in the protocol setup. So the hints and tips there would be to make sure your plate is sufficiently warm prior to addition of your culture into the plate. And there's some extra details of how to ensure that that's the case in the protocol that you can find on the website.
Great, thank you. Next question, how do I avoid cells growing in 2D in the embedded no base assay format?
Okay, so the converse is true here. Rather than keeping your plate very warm for when you're spotting those domes, here you just need to make sure that the plate's not too cold, so it's not pre-chilled for long periods of time on the cool sink, as we describe in the protocol. And speed is of the essence here. You need to start dispensing your organoid matrix gel cell suspension into your microtiter plate rapidly, because they will start sinking the slower you are. But again, extra details can be found in the protocol.
Great, thank you. All right, and like I said, we have time for a few more questions here and then we'll wrap up. But this next question asks, what concentration of matrigel should I use?
Okay, great question. This would really be cell type specific, but what I can share with what we've done with our validation, we have found that 50% matrigel has been sufficient to grow and differentiate the organoids that we've tested, as I said, in our hands. And indeed, some customers have reported that 50% also works in their hands, and they've looked at, as shown in the webinar, sort of iPSC-derived organoids, so brain organoids. There's been examples of lung organoids as well. Yeah, but my recommendation was to do a titration, but 50% is what we've shown works pretty well.
Great. All right, next question. Can I use an alternative ECM to matrigel?
Yeah, I guess this is what everyone wants to do and know whether it's possible. So all of the validation we've carried out here has been using Matrigel. A lot of protocols describe the use of Matrigel for organoid formation, growth, and maturation. We haven't validated alternatives to this, but there are vendors that do supply alternatives now. And what I would say is that give it a go, but just be mindful all of the validation and the supporting data has been generated with Matrigel. And I think as long as the alternative is optically clear, I think you're off to a good start with testing an alternative.
Great, thank you, Kalpana. All right, next question. Can I image my assay plates at 10x rather than 4x?
No. And I guess perhaps why you might want to use 10x is to provide you with that extra image resolution. But the downside there is that you've narrowed your field of view. So if you have very few organoids in your field of view, you're not going to, or you'll potentially miss the ability to capture them. So we're providing this extended field of view with a 4x image capture, and hopefully you can still get the resolution you're after, and the zoom in function as well will allow you to really dive a little bit deeper to look at some of those structures or morphological features in more detail.
Great, thank you. Now, if someone is growing spheroids in a Matrigel dome, is spheroid module or organoid module better?
So we would recommend, if you're working with organoids, to go down the organoid route of the supported software that's provided on the IncuSite. But if you are using spheroids as well, there is a IncuSite module that does support spheroids that are embedded in Matrigel, and that's called the embedded spheroid module, and it has a similar acquisition to the organoid module, but it is more optimal for spheroids, or specifically the visualization of spheroids in that gel. So we've got different visualizations of organoids and different visualizations of spheroids. So if you go down the route of the appropriate model on the IncuSite, you're in a good space.
Great. Now another question here. On a polymerase gel base, do you need to embed organoids in 50% Matrigel?
Yeah, so the protocol here is really allowing you to grow organoids that are embedded in an extracellular matrix. So they are distributed and sitting in different focal planes in the Z direction. So my answer to that question is yes, you would need to embed them. If you were to seed them just on the base of the matrix, it's a slightly different model. They're in a relatively narrow focal plane, in the same way that one of our spheroid applications work. Whether your organoids will actually form, grow, and differentiate just sitting on a base of matrix, I think you'd have to ascertain yourself. But we are supporting the culture model that uses organoids that are actually embedded and distributed within an extracellular matrix, not sitting just on top of one with media on top.
Great. Well, thank you, Kalpana. We're going to wrap up with this final question here. And this question asks, are there any minimum and maximum organoid size that is recommended for the analysis to be successful? For example, too big organoids are too difficult to segment.
Okay, so I think with the examples I've shown and our experiences with hepatic and pancreatic organoids, they grow quite large, up to a millimeter and a half. And the software hasn't struggled with the size of organoids in that range. I would say in the other direction, if they're too small and they have a similar size to, say, debris within the culture, the IncuSite segmentation algorithm may well struggle. So I think anything hovering around, I would say, less than 50 microns, it can be a bit challenging.
Great. All right. Well, that will conclude our session for today. So thank you, Kalpana. Do you have any final comments for our audience?
No, except thank you very much for joining, and I hope you found it insightful. And as I said at the end, if you wanted to find any more information, both on the InQ site, the associated software modules that support this organoid workflow, and indeed details of the protocols that were described, they can all be found on the website, and I think the links were also at the end of this presentation. So thank you for listening.
Well, thank you again, Kalpana, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until January 23rd, 2025. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.